Hey guys, this is Kendall Terry, and this is a look at the sensory system in vertebrates. Primarily is what we're going to focus on. So if we look at the sensory system, the first question would be, what are the sensory receptors? And there's two types that we can find. There's the exteroceptors. The exteroceptors are receptors that sense stimuli that arise from the external environment. So this would be primarily what we're going to focus on with this lecture, of looking at pressure, touch, hearing, vision, taste, smell. Those things would be what the exteroceptors would be for. And then there's the interoceptors, and those sense stimuli that arise from within the body, and this is how your body tells what's going on internally. So you always want that going on to help with homeostasis, because if you don't know what's going on inside as the environment changes, you won't be able to keep up with it. So what are the three classes of receptors that we're going to be dealing with? There's the mechanoreceptors. These are stimulated by mechanical forces such as pressure, and this comes into touch, hearing, and balance, and we're going to actually focus on all three of those in this lecture. And then there's the chemoreceptors that detect chemicals or chemical changes, smell and taste. Those receptors are not necessarily in the focus of AP biology anymore. And then there's the energy detecting receptors. They react to electromagnetic and thermal energy, and that would be the eyes. And then thermal receptors, like the pits in vipers, the pits that detect thermal imaging and that sort of thing. The four-step process of conveying this information, so you've got some kind of stimulus that takes place, and then there's a transduction of the stimulus to a receptor potential in the sensory receptors. So not only does there have to be a stimulus, but that stimulus has to be enough that it actually trips a receptor. It trips the receptor potential. Otherwise, you don't even really notice that. Then once that has been reached, there's a transmission of the action potential to the sensory neuron, and then finally there's an interpretation of that stimulus in the central nervous system. So depending on what that may be, that could be something that's being processed in the brain. It could be something that's being processed as kind of like a reflex reaction. We already talked about those with the nervous system, and those don't necessarily go all the way to the brain to be tripped. So how do we sense pain? We sense pain with the nociceptors. So these are receptors that transmit impulses perceived as pain. And then you have the transient receptor potentials, and these are the TRPs. These are ion channel proteins found in nociceptors, and they detect hot and cold. The hot responds to the chemical capsaicin, which is interesting because that's also the chemical that we find in chili peppers. And that's one of the reasons why chili peppers are perceived as being hot, is because they have the chemical that your body deals with as hot. Now, it's also the same chemical that your body will deal with when you're touching something hot. So if you're eating it, technically that chemical is giving tripping those nociceptors. And then you've got the cold, which responds to menthol, which is the same chemical that we see in cough drops. So you've got a sore throat. One of the things that's common to take is those mentholated cough drops. When you put those in your mouth, you get that cooling sensation. Well, why? It's because menthol is a chemical that your body already responds to, that it responds to cold with. So it's being used in the drug market, I guess you could say. And then there's thermoreceptors. The thermoreceptors are similar to nociceptors, and they also contain the TRPs. There's two types. There's cold receptors that are immediately below the epidermis, and there are numerous. There's numerous of cold receptors right there below the epidermis of your skin. And then you've got the hot receptors that are slightly deeper in the dermis, and they are not as numerous. So how do we sense touch then? Well, there's four main cells involved in touch, and you can see these on your screen right here. The first is called Merkel cells. Merkel cells are up near the surface of the skin, and they sense touch pressure and duration. So not only this is touching, and I feel that pressure, but these are also tripping a duration. So the longer I hold that on there, that is being perceived. You can feel that duration at that same time. Then you've got the Meissner corpuscle. Now, the Meissner corpuscle is found in a lot of hairless skin, and they're involved in sensitive to fine touch. So areas like the palms of your hands, the soles of your feet, your lips, areas of your body that are hairless. And you think about those areas, those are a lot of times considered to be very sensitive areas, and it's because of the Mesner corpuscle. Then you've got the Ruffini corpuscle. These are also, we consider them located near the surface of the skin, although they're a little deeper than the first two, and they are sensitive to touch pressure and duration as well. So you've got two, and this helps you understand how intense that pressure is. So you've got the Merkel that sits near the surface, that those are involved in touch pressure, but then you've got the Ruffini that sit deeper in the skin, although they're still close to the surface, they're a little deeper than the Merkels, and they also deal with touch pressure and duration. So they're dealing with that same response as the Merkel, and as those two are sending signals, your central nervous system then is figuring out whether this is something that is a problem or whether you need to deal with it, so on and so forth. And then there's the Pacinian corpuscle, and these deal with these are pressure-sensitive phasic receptors deep below the skin in the subcutaneous tissue, also dealing with that. These are the really deep ones. So if something is tripping all of these, then you know that that's probably an intense pressure, possibly something that you need to get away from. So your body has to deal with these as it goes. So that's a lot of the touch and pressure skin stuff. Let's move on now to how do we hear? Well, hearing is the detection of sound waves. And actually, this works better in water than in air because water transmits pressure waves more efficiently. And because of that, what we find is once we get into the inner ear, because we had to adapt to living in an air environment, and we know that sound travels better in water. So what you find evolutionarily in the development of the ear is a liquid-type environment that helps us transmit and understand the pressure changes, in essence, the sound waves. In fish, so if we go back to those very early vertebrates to see how this kind of development took place, we would go to vertebrate fish that have a lateral line system. Now this provides a sense of distant touch, so kind of like hearing for us, if you're out in the woods and something's coming up on this side of your body over here, you're going to hear it, and you're going to know it's coming from this side. Well, underwater, same thing is if something's coming close to you over here in the water, it's going to create a pressure change in the water. So that pressure change enables the fish to know that something's coming from this side, and if it's coming from this side, more than likely, evolutionarily, survival would say, get away from it. So go the other way. And this enables them to sense objects that reflect pressure waves and low-frequency vibrations. And this is what the system looks like. And you've probably, if you've ever caught fish, you've probably caught fish that have this lateral line in them that help them to understand the pressure. So you can see here that lateral line actually is made up of little openings, and they're connected by a canal. And in that canal, you will find these hair cells. And this hair cell has cilia with a cupula around it, a cupula kind of like a gelatinous type structure around it, and that air that around it then will allow the liquid to move the cilia and that hair cell, and how they're moved will trip the nerves that are sent, and you can see the connection here to the nerve, and this lateral line organ will help understand then when something's getting close, that distant touch. So if something gets close, I'm going to go away from it on that side. That way, as a fish, you don't get snuck up on. Let's look at the vertebrate ear then. So the vertebrate ear is made up of three main areas. You've got the outer ear, which is the ear canal, eardrum, tympanic membrane. The middle ear, which is the malleus, which you've also maybe heard it called the hammer. The incus, which you may have heard as the anvil. And the stapes, which you may have heard called the stirrup. And then you've got the inner ear, which if you look at the inner ear, that's the oval window, the cochlea, and that cochlea then is connected finally to the round window, which you'll see here. And when I mention that later, that's why I'm drawing attention now, is you'll know where that is right there. So how do we hear? Well, there's first the vibration in the air, vibrate the tympanic membrane. So something's coming in, it goes into your ear, and it causes that tympanic membrane to vibrate. These vibrations cause the middle ear bones to move as well, because you can see here that they're connected there. So as they move, then you get that vibration sent through the oval window, and that pressure waves are then transmitted through the cochlear. And you got here until it approaches the round window, and that round window here to where it will exit the cochlear. So then the sensory apparatus inside of the cochlear consists of a basal membrane, with hair cells. So here's the basal membrane. Here are the hair cells. Those hair cells obviously connected to the auditory nerve right here. So as those hair cells in here are moved as those vibrations come through, they will trip the auditory nerve. And the deeper the voice and the higher the voice will cause different amounts of vibrations to go through that cochlear in different to different locations. So one may only go so far, and your brain will understand that that's either a deep voice or a light voice or whatever may be the case. The other goes a little bit further, and you get a different sensation in your brain to recognize the different types of vibrations. Now this is, you can see up here, it's got this membrane that's called a tectoral membrane, and all of this together that you see on here is all referred to as the organ of Corti. The organ of Corti connects all of this kind of how do we hear to get up together in understanding those vibrations going through the cochlear. The last thing I want you to look at is how do your ears help us detect position and movement. So here we've got position. You've got these hair cells in the cochlear, and those hair cells have a kind of like gelatinous matrix around them that's made up of kind of like a liquidy type material that can move. So as you move the position of your head, if it's down like this, that fluid is going to go forward. It causes the hair cells to go forward, and it helps your body perceive that you're moving forward. Same with if you go back, okay, you get that movement of that gelatinous material. You can also get this with side to side, although with side to side, we use a different structure. You can see here, right here at what we call the ampullae. In the ampullae, you have this kind of this indention, if you will, that sticks up, and with those hair cells around it, you got the capula around it. Notice this is the cupula. Notice this is very similar to what we saw in the lateral line system. The liquid here, the endolymph, and as you move your head side to side, that endolymph has to flow through as you move, and it causes that cupula to move, and that trips the hair cells, and that helps you understand the movement of your body. So all of that goes into how we understand the environment, how do we hear, and how do we perceive movement and position.